Hi, everybody. Thanks for having me. Last time I was here, I was giving a talk that said what ME/CFS experience can teach long COVID folks. So this time I thought I'd flip it. It's been a year later. What have we learned about long COVID that's going to help ME/CFS folks? How's that for turn that dime? So that's what I'm going to do. So here we go. See how I do. I'm sorry, you're right. I'm paying attention to my earlier instructions. So it's been an amazing conference these last 2 days. We've learned so much. It was perhaps the best conference ever. Some really fantastic lectures, and I feel like I'm coming away with new knowledge, which should be very exciting and very hopeful to the patients in the room because it's very exciting. Part of this is that long COVID has pumped so many new investigators into the field, and I mean a critical mass of new investigators that are making great progress. You heard that lecture this morning, Dr. Wurst, who has a Nature article on a muscle and post-exertional malaise. It's extraordinary science, really phenomenal science. But everyone, one right after another, these last 2 days have given us some amazing insights. So a critical mass of investigators, incredibly important to the whole field, including to our ME/CFS field. So I'm going to try to do a little something with all of that. So one of the takeaways is that ME/CFS and long COVID have far more in common than they have apart by a lot, say 80, 90%. The science will go on to figure out what that is. But the common mediators of persistent illness are there. And as we learn for long COVID, things that work, they will hopefully and should apply, if you group the patients correctly, to ME/CFS. So that's exciting. That's really exciting because there's a lot of work going on all the way to clinical trial in long COVID. And so these common mechanisms are things like viral reactivation, immune dysregulation, dysautonomia, and so on. And you have great speakers today talking about all of that. We have a study underway. It's a CDC study called COVID Up, Understanding the Post-Viral Phase, where we are having the opportunity to look at long COVID patients and compare them to the 750 patients that were in the MCAS study that Dr. Unger spoke about yesterday, which is great. They were just pre-pandemic, this amazing phenotyping work on a very large cohort of patients. So we designed a study that was very similar so that we could do that compare and contrast. So you'll be seeing more on that. We're more than halfway through, maybe 2/3 of the way through, and it's going very well. So I'm going to talk about that because I want to give you more clinical pearls. So what are the key observations in general between long COVID and ME/CFS? This persistent activation in the immune system, elevated pro-inflammatory cytokines, the autoimmunity subgroup, and perhaps something that will evolve with duration of illness over time, as it does in ME/CFS in long COVID patients, and that there's implications for therapeutics in all of that. The takeaway on this slide is that we have to design our studies to include the ME/CFS sick control group when we're doing long COVID studies. Not just because we're going to learn even more about long COVID that way, which is the intent of that funding, but because we'll be able to generalize that work over to ME/CFS and give the ME/CFS population labeled drugs that are for their disease too and not off-label. And that's incredibly important. And not everything's going to be drugs, but I'm just using that as an example. The role of viral persistence. With ME/CFS, and I've been writing this way for decades and decades, sometimes we talk about it, sometimes we don't talk about it, then we talk about it some more. But right now we're talking about it some more. Why? Because in long COVID, if you reactivate Epstein-Barr virus during acute infection, you're more likely to have long COVID. So now we're talking about what is the role of co-infection, of that other infection, at the time of risk to develop long COVID. And then the second part of that, what does it matter if you still have it now 6 months later and your COVID is apparently gone, but you still have reactivated reactivated Epstein-Barr virus? So this is a very strong area of research in the long COVID work, and it's extremely relevant to the ME/CFS work. And of course, in ME/CFS, we talk about other viruses too, as they are in long COVID, but it suggests there might even be a treatment that goes down the line of controlling viruses. That would be great. I was an HIV person who thought that we would never repair the immune system after it was so badly damaged by HIV virus. And what happened? We had people whose CD4 counts were 0 that went all the way back to normal and healthy just by getting rid of the HIV virus. So I'm a person who speaks a lot about having to push more than one button to get people well with ME/CFS. But if in fact there was a core central antigen driving the whole big mess, I'd like to be wrong. That would be great. So in long COVID, there is also an argument that COVID persists. Not necessarily in everybody. And the reason why we don't know this is that we don't have a biomarker that measures it very well. Viruses can go into protected compartments. They're not in the bloodstream. We can't access them to look at them. It's true in ME/CFS when we're looking at enteroviruses that restrict to the gut, and we can't measure them correctly in the bloodstream. So same thing in long COVID. What if COVID persists? Well, we published a paper, a little case series of 3 patients in January where a monoclonal to COVID, one of the treatments for COVID, actually completely cleared and reversed the long COVID course in 3 consecutive patients. And since then, I think there's 17 patients in this particular practitioner's practice that have recovered because they had access to these monoclonals. So we have worked long and hard to create a monoclonal treatment for long COVID at a time when all the monoclonals were no longer available, that they've expired and they don't work on Omicron, and all kinds of problems with the monoclonals. And we couldn't get any of the old monoclonal companies to work with us. But happily, things are moving forward. We have a new partner, and it's a product that covers the current variants as well as the prior variants. So I'm having a meeting this afternoon at 5 o'clock to keep that moving. Every week we move it, and it should be in front of the FDA in the next 3 to 4 weeks. So that's exciting. I'll let you know. That study will be over this time next year because we're driving it hard, so you can ask me about it. Autonomic dysautonomia, I'm not going to spend any time because you have David Systrom. Who could ever explain more about dysautonomia than David Systrom? But let's just say it matters, and it matters in both illnesses. So, years on when David talks. Chronic inflammation and oxidative stress. This is very, very relevant to both illnesses and a very treatable place, probably the most treatable place. So today you have Dr. Reneska and Dr. Rosenfeld speaking from our group, and then you have the European speakers talking about the ways we manage these complicated patients, removing the layers of the onion, trying to fix what we can fix. And that approach is very much focused on how to treat these things. But in this conversation, you have to respect that the blood-brain barrier matters and that the antioxidants don't cross all that well. And so there's some studies, I think Dr. Shungu has one that's underway right now with the NIH and ME/CFS, and we have one with Dr. Shungu as well in Gulf War illness, where he's demonstrated that NAC, N-acetylcysteine, crosses the blood-brain barrier enough to change the image, the way you can image oxidative stress. So when you're looking at antioxidants, think also how deep do they go? Endothelial dysfunction. Now this is something we're absolutely learning from the long COVID people. We talked a little bitty bit about it. There's 3 or 4 papers, some good papers from mostly Europeans. I can say no US papers on endothelial dysfunction in ME/CFS. But now that we realize how incredibly important it is in long COVID, all eyes are on the endothelium. And it's a great place to be looking. Why? Because there's direct and indirect ways that endothelial inflammation and endothelial dysfunction affect every tissue type in your body. And when you have an illness that presents with absolutely everything involved, you have to go to the circulation, go to the capillaries, go to the endothelium and say what's going on at that level. And so in this instance, let's see if I can use my pointer. In this instance, there's many different ways the endothelial can get involved, including direct release of pro-inflammatory cytokines, which we definitely know are elevated in both illnesses. But the much neglected cell, the one that no one ever looks at, the neutrophil, it's been floating around there being 30, 40, 50% of your white blood cells. It can become engaged in this because when it breaks down, creates essentially a net and creates a clot, a little microclot, grabs up some cells going by and creates a microclot. And these microclots have been documented to be very important in long COVID. But they're also present in ME/CFS, not probably as much, not as intensely, but there, and there's literature to support that now. So that really matters. So when looking at endothelium, it's in the arteries, it's in the veins, it's in the capillaries, the arterioles, the venules, capillary beds. It's also in the lymphatics. So these are important because if you go back to this, COVID directly binds to these two receptors. There's a receptor and a co-receptor, and there's the COVID virus, and it makes a localized inflammatory reaction at the level of the binding. And that makes tons of good sense, okay? But Epstein-Barr virus is also known to affect the endothelium and cause release of pro-inflammatory cytokines. So just because we know COVID does this doesn't mean these other viruses aren't important. And in general, viruses that circulate in the bloodstream have a lot of exposure to the endothelium, so they can cause problems. And both the blood vessels, the veins, the arteries, and the lymphatics have these funny little tiny slippery barrier called the glycocalyx. And it's the very first thing to be injured in cell wall injury in the blood vessel. And once it's broken, once it leaks, once it's causing problems, then you can imagine how the cascade of events might then follow. And these lymphatics are everywhere. They're everywhere. That's your white blood cell, transports itself here, there, and everywhere through its own vascular system that we hardly ever talk about, but it's really important. And it's important in the brain where there's something called the glymphatics. Believe it or not, with all the amazing anatomical discoveries of the last 200 years, no one even noticed the glymphatics until the last 20 years or so. But they exist and they do the same thing. They follow the blood vessels of the brain. And Dr. Partnam was here just the other day talking about an upside-down circadian rhythm for sympathetic and parasympathetic nervous system and suggested that the sympathetic tone, the cholinergic drive at nighttime, was reversing and diminishing the flow of the glymphatics in the brain, which makes lots of good sense and would reduce your ability to recover during the night and have brain repair and deliver the cells that you need in the area. So it's an important thing. So that brings us to neuroinflammation. Neuroinflammation is a thing. We've documented it very well in ME/CFS. It's now well documented in long COVID. And it's an amazing good target for potential treatments. So I know that there's a low-dose naltrexone trial underway in a multinational way, which is very exciting. But other potential anti-inflammatory treatments are plausible. This was an important paper out last month in long COVID. They could distinguish brain fog people from not brain fog people by blood-brain barrier breakdown. They could show the endothelial blood-brain barrier, and they picked out correctly all 11 patients out of these 22 patients by this kind of imaging, DCE imaging, that measures leakage of the blood-brain barrier. That's important because your blood-brain barrier is keeping all this systemic stuff away from your brain. You're not supposed to be swimming in tumor necrosis factor, and all the hormone levels that are on this side aren't the same on the other side. And it's terribly important that these two compartments be kept very separate from each other. And so that's an exciting development and gives us, again, the endothelium as a really important target for treatment in both illnesses. It helps us focus. I'm not talking about metabolic dysregulation because you have Ron Davis as your last speaker today, and he's talking about metabolic dysfunctions. So, but this is, a hot area and a hot topic in both fields. And there's a lot of different potential ways to treat metabolic dysfunction. I will say in our own practice, you'll hear more about it later, it's a very personalized approach. Everybody's different down this line. And if there's a way to measure things, you do a much better job of getting them right. And I don't want to rain on the parade of the first speaker after lunch. Am I doing okay on time? Yes, I'm good. Okay, a couple more minutes. I just want to spend 2 slides on glycocalyx. I just mentioned about this slippery little fuzzy thing that holds all the antioxidants in this nice little barrier here and lets the cells slide through and does all these important things that breaks down in both of these illnesses. Is there some way to repair the glycocalyx and encourage vascular repair? One of the best things about the human body is it loves to repair. Otherwise, we wouldn't make it to 10 years old. You know, it's a very reparative place. It's a regenerative place. There's a lot of chance to recover. Even very broken things can recover. So, and broken endothelial cells should be able to recover or revascularize, which is another thing. You can actually grow whole new blood vessels when you need them in certain areas if they're the little ones. So there's a lot of different ways to go. But there's a lot of things that are broken in here, like the nitric oxide levels are off. You'll hear more about that from Ron later. And the lumen of this vessel is small. That can be from the cholinergic, from the sympathetic nervous system, but it can also be anatomic, which you saw, or we saw a little bit of yesterday in the bigger talk. So it's a very important area, and perhaps there are better ways to think about what to do about it than what we're going to do about it. But we have a clinical trial, excuse me, I'm going to jump over that slide for time, that will also be starting in the fall of this year. We have things ready to go using this particular supplement, which is proprietary, and I'm not recommending you use it until we do the study and tell you if it's great. But again, next year, this time next year, we'll let you know whether or not this is useful. But Arterisol, which is made by Calroy pharmaceutical company, is a nutraceutical. It's made from algae, and it has a slippery, slimy little internal product that absorbs, and they have some pretty decent data to show that it can go to the glycocalyx and help to repair the glycocalyx. That study will be using a nitric oxide supplement at the same time to repair the reduced nitric oxide levels. So it's sort of a phase 1 with one drug and then phase 2 with both drugs kind of study. So we'll let you know how that goes. But again, we're looking at both ME, CFS, and long COVID in that study. So lastly, what else do we learn? Boy, does money matter. I mean, this is like duh. But look at this morning, I went on clinicaltrials.gov and I pulled this down. In long COVID, there's 517 ongoing human studies. That's not all the lab stuff going on. This is just the stuff that's recruiting human beings into their studies right now. There's 180 clinical trials listed, and 71 of those trials are recruiting. Now, this is a new disease. It's only been around a few years. All this happened in the last few years. In the last 20 years, which is what it pulls out of the same math, there's been 294 human studies of ME/CFS that recruited humans, of which 64 are currently active, 105 clinical trials over all the last 20 years, of which 25 are currently recruiting. So that's a huge difference, and this is internationally. This is not the US numbers. This is everyone lists their stuff on trials.gov. But wow, money, less money. So keep it in mind that money matters, and that as an advocate, one of the things I would most like to hear is that ME/CFS is linking its bell to the long COVID money chain. Because if you could do comparator studies, you're going to move way faster than trying to start from scratch and get that kind of advocacy going that long COVID is having in this so-called post-COVID, post-pandemic era. So keep that in mind. So what are the therapeutic implications? There are a lot of different spaces. Actually, in this entire talk, we haven't talked about mast cell stabilizers. I hope you'll hear about it this afternoon, or the role of toxins, which are terribly important in mediating disease persistence. But there's a lot of different buttons that can be pushed, a lot of different trials that need to be done, and a lot of forward momentum from this conference to be collaborative, organized, networked, and move it forward. So again, take some hope from the amazing organizers of this meeting and all the people that came to try to make that successful. And that's pretty much that. I had one thing I wanted to share with you that, of course, my screen went dead. Sorry, my little password moment. Yeah. Back to microclots, just some practical talk. You clot more when you're dehydrated, so don't get dehydrated. These are diseases with low blood volume, and dehydration is as common as it can be. You clot more when you have inflammation, and these are diseases of inflammation. So don't make it worse by eating inflammatory foods and promoting inflammation. A lot of refined sugars and refined carbs promote inflammation. So there's things you can do in your own diet, taking away inflammatory things and putting in anti-inflammatory things that can actually have day-to-day real help. There was a paper out 2 months ago or last month that xylitol, my favorite fake sugar, induces microclotting. Great, because now, I mean, there's really no fake sugar that's healthy sugar. But xylitol, your body makes xylitol, so we thought that was a good one. But now there's some data saying it promotes atherosclerosis and it promotes microclots, so be careful on things like that. And there's things you can do to try to make that better, which is stay very well hydrated, and eat anti-inflammatory foods, and eat and the things with omega-3, and then be careful with your nutrients like vitamin D, which is anti-inflammatory in the brain, and healthy fats and healthy grains and that sort of thing. And finally, we have a podcast in the top 1% internationally of downloads, not just in the health category, but in all categories, because people are so hungry for pearls of wisdom, which is what this is, pearls of wisdom, for people with long-term chronic inflammatory illnesses like this. It's called Hope and Help for Fatigue and Chronic Illness. Hope and Help for Fatigue and Chronic Illness. And there's a lot of them, and a new one comes out every Tuesday, and it's been very well received. We'll be tapping some of you speakers to be guest speakers on our podcast. Thank you.